Immunology Insights, Basics and Beyond, Chapter 29, Hypersensitivity and Allergic Reactions. Marcello here. Welcome back to Immunology Insights, Basics and Beyond. In our previous chapter, we explored the life and legacy of Albert Sabin and how his oral polio vaccine helped bring the world to the brink of polio eradication. Today, we switch gears and focus on what happens when the immune system goes overboard, leading to hypersensitivity reactions, from common hay fever to autoimmune diseases and even life-threatening anaphylaxis. So let's talk science. But what are hypersensitivity reactions? Hypersensitivity reactions occur when the immune system responds inappropriately to otherwise harmless substances or even to components of the body itself. British immunologist Philip Gell and Robin Coombs introduced a now widely used classification system that breaks hypersensitivity down into 4 types based on their underlying mechanisms. Type 1 hypersensitivity, immediate allergic reactions. Type 2 hypersensitivity, antibody-mediated cytotoxicity. Type 3 hypersensitivity, immune complex mediated reactions, and type 4 hypersensitivity, delayed type, so cell-mediated reactions. Each type involves distinct immune components and manifests differently in clinical settings. So let's take a closer look. Let's start with type 1 hypersensitivity, immediate allergic reactions. The key players are IgE antibodies, mast cells, basophils, and histamine. The mechanism is as follows. When first exposed to an allergen like pollen, peanuts, or pet dander, B cells produce IgE antibodies specific to that substance. These antibodies bind to mast cells and basophils, priming them. On subsequent exposure, the allergen cross-links these IgE antibodies, triggering a rapid release of histamine and other inflammatory mediators. Clinical examples include hay fever, so allergic rhinitis, which manifests in sneezing, nasal congestion, and itchy eyes, or asthma, which means bronchoconstriction and inflammation in the airways, and of course anaphylaxis, a severe systemic allergic reaction requiring immediate epinephrine treatment. Treatment strategies for hypersensitivity type 1 include antihistamines, of course, to block histamine receptors, but also corticosteroids for inflammation that reduce inflammation and immune overactivity by blocking immune cell pathways like cytokine production and T cell activation and others. Then we have allergen immunotherapy, so desensitization, and epinephrine autoinjectors for emergencies. Now let's move on to type 2 hypersensitivity, so the antibody-mediated cytotoxicity. Here the key players are IgG, IgM, the complement system, and natural killer cells, so NK cells. Mechanism is as follows. The antibodies bind to host cell surface antigens, marking them for destruction. The complement system is activated, leading to cell lysis, while the natural killer cells may also mediate killing through ADCC, which means antibody-dependent cellular cytotoxicity. Clinical examples include the hemolytic disease of the newborn. Here, maternal anti-rhesus factor antibodies destroy fetal red blood cells, leading to severe conditions. Or myasthenia gravis. Autoantibodies block acetylcholine receptors at neuromuscular junctions. And Graves' disease. Here, autoantibodies stimulate the thyroid gland, leading to hyperthyroidism. Treatment strategies include plasmapheresis to remove the antibodies, immunosuppressive drugs, like for example corticosteroids or rituximab, and thyroid hormone modulation in the Graves' disease. Next, we have the type 3 hypersensitivity, the immune complex mediated reactions. Here, key players are antigen-antibody complexes, the complement system, and neutrophils. As for the mechanism, immune complexes form in the bloodstream and become trapped in tissues, especially in small blood vessels, kidneys, joints, and skin. This activates the complement system, attracting neutrophils that release enzymes, causing tissue damage. Clinical examples include systemic lupus erythematosus or SLE, Immune complexes target DNA and other nuclear components. Or rheumatoid arthritis, or RA. Here, chronic joint inflammation driven by immune complexes causes severe damage. And serum sickness, a response to injected foreign proteins, for example antivenoms or old rabies sera. Treatment strategies include non-steroidal anti-inflammatory drugs or NSAIDs like ibuprofen that block COX-1 and COX-2 and thereby alter prostaglandin production, and again corticosteroids for inflammation, or disease-modifying antirheumatic drugs or DMARIDs in rheumatoid arthritis, and of course biologics like anti-TNF antibodies. Last but not least, we have the type 4 hypersensitivities, or the delayed type, so cell-mediated reactions. Key players here are CD4 and CD8 positive T cells, macrophages and cytokines. In regards to the mechanism, unlike in the first three types, this response is not antibody-mediated. Instead, T cells recognize antigens and release cytokines that activate macrophages, leading to inflammation. This reaction typically takes 24 to 72 hours to appear. Clinical examples include the tuberculosis skin test or Mantoux test. It shows prior sensitization to Mycobacterium tuberculosis. Or contact dermatitis, a rash caused by poison ivy, nickel or latex exposure. And also type 1 diabetes. Here, CD8 positive T cells destroy insulin-producing beta cells in the pancreas. As treatment strategies, for example, topical steroids for mild dermatitis are used, or systemic immunosuppressant for autoimmune diseases, and of course insulin therapy in type 1 diabetes. This was just a very short overview of the 4 types of hypersensitivities. More details might be covered in other chapters of this podcast. To conclude, hypersensitivity reactions underscore how the immune system designed to protect us can sometimes become a source of harm. From itchy eyes to organ damage, the consequences vary widely. By understanding the mechanisms involved, we can better tailor treatments, improve diagnostics, and in some cases even prevent immune overreactions altogether. In the next chapter, we will take a step back into history to meet one of the early visionaries who shaped this field, Clemens von Pirquet, the Austrian pediatrician who coined the term allergy and helped us to see the immune system in a new light. Before closing, I have a small request. If you enjoyed my content and want to hear more, like and subscribe. It's a small step for you, but it helps me a lot. So see you next time and thank you for listening.